All right, everybody. Welcome back. I'd like to introduce Siwen Liao. She's a second-year undergrad at the University of Virginia, studying statistics and physics, and she's going to discuss the art of brain data in ASD subjects, celebrating neurodiversity through aesthetic data visualization.
Yeah, thank you guys for your time. I'm currently an undergrad, as introduced, and I'm doing research with Professor Van Horn, and this is what we've been working on this year. So we're going to be looking at brain data in ASD subjects. So just as an introduction, when we talk about ASD, we mean autism, and it's just a shorthand. I'll be using it throughout this presentation. So currently, autism affects roughly 1 in 36 children, according to the CDC. This number is increasing, and a large reason is that we know a little bit more about like what ASD looks like. Actually, 20 years ago, it wasn't believed that girls could have autism. So this increase of awareness is what is increasing these numbers, and these numbers will probably continue increasing. So it is a very prevalent disorder. We are also mainly going to focus on the gender gap in autism. The current ratio between males to females in diagnosis is 4:1. And not only is there a gender gap in the volume of diagnosis, there's also a gender gap in like the average age of diagnosis. So we can see here that the age that females are diagnosed is greater than the age that males are diagnosed. And whether this difference is significant, research papers sometimes disagree on, but generally this is a trend that contributes to the gap. And like all clinical disorders, earlier diagnosis is more important, so this is also like important. And there's theories in psychology research as to why there's this gender gap, and one of them is the female protective effect, where females tend to mask symptoms more than males. So they require heavier symptoms to be able to be diagnosed at the same rate. And we touch on this because a lot of autism diagnoses tests are behavior-based. And in this project, we're looking at brain volumes, which can be correlated to behavior, but it is more of a quantitative approach. And hopefully we can potentially identify biomarkers in brain volumes and use that as diagnoses to find another way besides behavior-based tests. So the two goals of this project, we had sort of two parts going on at the same time. The first was the data analyses. We used multivariate statistics. We'll be talking about that first. And then concurrently, as we were doing that, we also 3D printed a lot of brain models because it's a cool way to show complex data in a more approachable way to increase awareness of this gender gap in our findings. So we're going to first talk about multidimensional scaling. This is an unsupervised view. So what we're trying to look at is the natural structure of data and whether the data sort of sorts itself into groups. So we don't put labels or we don't sort the data into groups beforehand. We kind of just see how they sort themselves. And here, I'll show a couple graphs, and it'll be 2D because according to the scree plot, which is a method for determining the optimal number of components useful to describe the data, we can see that the elbow is at 2, so we're going to be looking at 2 dimensions. So our first plot here, we can kind of see that this plot, the differences is mainly due to gender. Generally, guys have larger heads than girls, and we can see that difference here where regardless of whether the patient is ASD or neurotypical, the guys tend to have larger heads than girls. So we kind of get rid of this effect by normalizing the data by dividing by intracranial volume, and we can see now that you can't really see the gender divide anymore, and rather we're just looking at the diagnoses effects. And we can see that the patients with ASD are more widely dispersed. You can kind of see on the outer rim, it's mostly ASD observations, whereas the control observations are more clustered in the center. And we continue to visualize the data using a dendrogram, using the word distance. And we go back to using unnormalized data, and we can kind of see that in that it's mostly divided by gender, where the yellow and the green are girls, and the red and the blue are guys. And you can kind of see that divide in the labels on the bottom, not the branches. So we can also kind of see that the green and the girls clusters a little bit more commonly on the left than the yellow. So this does imply maybe there is some gender difference in diagnoses. So now that we've taken a look at the natural structure of the data and how it groups itself, we now turn to linear discriminant analysis to see how well we can classify the data and also use this to potentially identify the top regions that contribute to the differences in brain volumes between groups. So the idea of linear discriminant analysis is we're trying to find the best optimal linear combination, so essentially the best line, to maximize the separation between the different groups, but minimize the inside of each group. So increase variance amongst the groups and decrease variance within the groups. So we can see here that after we found the top eigenvectors, essentially the directions of the lines, we can project the data onto these new dimensions and it'll form like separated groups. So here we projected it onto the top two eigenvectors, so the top two lines, and you can see that it forms nicely with 4 groups. And if you want to take out your phones, this is a QR code to a 3-dimensional projection with the top 3 eigenvectors, and it's more of an interactive plot. So you can kind of toggle around and you can see like the different groups from different sides. Okay, awesome. So we're gonna now look at the different eigenvalues and what they represent. So the first eigenvalue, as we can kind of already infer from the multidimensional scaling analysis we did earlier, was that the first eigenvalue, the largest contribution to the variance, will be gender. And we can kind of see from the legend that the females are on the right and the males are on the left. The second eigenvalue, which contributes to the second most variance, is the ASD diagnoses, and we can see that they're separated here. And then the third eigenvalue, which still had a significant contribution, but less contribution than the other two, was the interaction between diagnoses and gender. And we can see here what's interesting is that the girls diverge further than the guys, and that's something that can be looked into. So similar to multidimensional scaling, we want to adjust by intracranial volume just to be able to see the effects of ASD more without having gender take up so much of the variation. So when we project the first 2 discriminants after dividing by intracranial volume, we can see that now the gender effects are removed, although it is interesting that the males are more like widely spread and the females are more clustered in the center. And we can see now, because we divided by intracranial volume, the eigenvalue that was originally gender has collapsed, and we can no longer really see the gender differences. And the second eigenvalue still remains as the ASD diagnoses, and we can see that it kind of separates itself into gender, but also there's variation between the ASD and the neurotypical groups within each gender. And now for the third eigenvalue, the interaction value, we can see that more of the variance is explained by this eigenvalue, and we can see that the males have a very similar distribution, but the females tend to differ a little bit, which is really interesting. And we can see the significance of these eigenvalues by calculating the Wilks lambda and then approximating it using the chi-square distribution to find the significance. And we can see that both of these three discriminants for each respective process was significant. So that was the statistical analyses, and the other part was the printing brains. So we printed a lot of brains. We printed over 300, and I will be talking about some of the difficulties we had along the way, such as dropping the brains on accident. Oh, it says can't play. Oh, okay. Well, it was a video of just the 3D printer printing the brains. Hopefully the videos, the other videos work, but we'll see. So here we kind of I took pictures of the process, and on the right we were given data files of the brain surface models, and we were given the data set. So I had to cross-reference because the surface models were labeled by their ID number. So I had to cross-reference and identify sort of what group each of the surface models belonged to. And halfway through, I kind of realized that it probably would have been smarter to run it through some sort of algorithm or automate it, but I was already halfway through. So I just created this spreadsheet kind of organizing everything. And we had 4 groups. As we saw in the analyses, it was male ASD, female ASD, male control, and female control. And each of the groups were printed in a separate color. So once they were printed, we stored them. They're still sitting there in Gilmore Hall in the cabinets. But we had to label them with their ID number. And you can kind of see here in the top left, the green ones are not as organized, and the labels are a little bit wonky. That's because I did drop those groups, and I had to look in Prusa Slicer and see sort of the crevices and try to identify which brain matched with which crevice and try to match it back to the ID number to get everything sorted. That was unfortunate. And then you can see the middle one, the brain slightly looks deformed, and we hypothesized that this is because the file just didn't process correctly, because we cross-referenced with our analyses and they were extreme outliers, so we just ended up removing them from our test. So yeah, essentially we just imported the files into Prusa Slicer, sliced them, stuck them into the 3D printer, and then they just printed for a couple days, and we labeled them and stored them. And hopefully we'll be using them to create our 3D physical data visualization. Okay, so now for our results. So we can see here that these are the top 20 brain volumes that contribute to the first discriminant, which, as we saw, represents gender. And this does correspond with previous research, because the top 3 volumes, there has been shown some differences within gender. And then here's the top 20 features contributing to the second discriminant, which is the ASD diagnoses. And we can see that these are more related to social and motor processing, which also supports previous research on ASD. And then we can see the top 20 features contributing to the third discriminant. And this one is more interesting because there hasn't been that much research done on it, but we can see that the top regions contribute a lot to emotional processing. And that's something that can be studied further, because evidently we saw there were some differences in how ASD manifests itself in females versus males, and looking at those brain regions, we can perform further research as to maybe like why. And then what we did was we took the Distro atlas, which is a common atlas in neuroscience that identifies the regions, and we took vertices from those and the corresponding names, and we matched them to the names of the regions of our linear discriminant analysis weights, and then we were able to color the vertices based on the severity of their weights, which essentially is how much they contribute to a specific separation. So on the left, it's the first discriminant, and on the right, it's the second discriminant function. And we'll go further into detail about what the different colors can mean. Okay, well, it's okay. I guess I'll go back then if we can't see the video. So on the left here is the first discriminant, as we mentioned, which is tied to gender. And the areas that are sort of highlighted are the yellow ones and the blue ones. But it's mostly in the face processing regions, which is interesting. And there has been like a little bit of research in differences in face processing between males and females. So this is what that represents. And then we can see on the right, this is the second discriminant. So it's the ASD versus neurotypical mapping. And we can see that the language processing areas are yellow, which is interesting in that potentially this could support the idea that there's language processing differences between individuals with ASD versus neurotypical individuals. Yeah, this slide kind of reinforces what we just touched on. Formally, it's called the Wernicke's area. And then for the third discriminant, which is the interaction between gender and ASD, we can see that the regions that are highlighted, along with the top 20 regions that we just looked at for this discriminant function, is that it relates to social processing, sensory integration, and emotion regulation. And this is what is most interesting and can be touched on further. So on the right, there's another 3D graph. Essentially, what we did was that we took the 2D graph on the right and we turned it into a three-dimensional graph by raising each of the observation by their age. So it'll mostly be flat, but it just gives a little bit of dimension since we do have 300 brain models, and we kind of want to layer them somehow. So that's the idea behind what we'll be doing with the printed brain model so far. And for future research, we can analyze the proportion of individuals from each of the groups just to see how more in depth the data like categorizes itself. And then we can also use the linear discriminant model to build predictive classifiers for clinical use. This is what we touched on earlier with the idea of potentially moving away from behavior-based tests to more quantitative tests to potentially surpass the female protective effect. And then we can, as we touched on, can investigate whether the linear discriminant, the third one, the pattern, the interaction between gender and ASD could serve as a biological marker and using that for diagnoses. That is all. Thank you for your time. And I would take any questions.
Yes, maybe you mentioned it and I didn't process it in simulated in some kind of like 3D model on the computer environment, kind of like a video programming or game programming thing, what do the physical models contribute? Is it more for like visual aesthetic purposes, or is that kind of fed into a different processing thing, like for the 3D modeling? Does that make sense?
Yeah, so you're talking about like the large overall visualization with the brain models?
Right. So like,
Yeah.
why take the time to print 3D print so many if
Yeah.
they're going to be simulated in a 3D program? I guess.
For now at least, like the QR code ones, they're just dots. But the videos that didn't show were more like the big 3D models, where we inputted it into Houdini and sort of visualized they like turned. It was pretty cool. So I'm sad that I couldn't show them.
Is it on the GitHub? Your GitHub or anything? On the video?
No, but that is a good idea.
Thank you so much. That's
Yeah. But so mainly the brain models for the larger visualization is for aesthetic purposes. But I mean, if you look closely, there could be some like sizes. Like at least when we printed out, I noticed there were like different sizes of brains, so that could contribute to the meaning of it. But mainly it's for aesthetic visualization since they're relatively small, so you wouldn't be able to see like the intricacies of it.
No, I think it looks cool. I guess
Yeah.
I wondered if I missed some kind of thing where you take 3D dumps and then move it to something else, and that is the like 3D larger visualization. Then.
Yeah.
Cool. Thank you so much.
Yeah. Of course. Yeah.
I have, well, many questions. First, awesome subject. Love the
Thank
subject.
you.
Quick one, just in terms of the variation that you see in the volume, the brain
Yes.
volume, has that been normalized by age?
That has not been normalized by age. Our data set is focused on children, so the age range is a little bit small, and I noticed that when I was creating the 3D model visualization. It was actually in months, so it was like 100 something, like 200 something, and I was like, well, what does this mean? And I had to ask my professor, and he was like, divide by 12, like it's in months. So the age range isn't super big, but it would be interesting to normalize it by age for sure.
The second question I have, I think, is probably more impactful. As you correctly identified, ASD is a diagnosis based on behavior.
Mhm.
And so to get an ASD label, especially as a female, we already know there's biases there.
Yeah.
Right? And so I guess my question is, I like to call this ground truthiness. Were there any steps you took to try to analyze that, like how that bias could affect your results in some way, or to mitigate that bias?
No, just because we were focused more on like the quantitative aspect. But I think that would be interesting in that the data possibly could be biased, in terms that if we compare this to previous data, there has been more data collected for males with ASD, and that could possibly hinder our results if we like apply it to perhaps an a linear discriminant model. Yes.
Awesome presentation. Really cool topic.
Thank you.
I wanted to geek out a little bit more on how you took the basically your data points and then mapped them to a 3D brain model.
Yes.
My understanding is that each part of the brain is like a designated ID to which you then, to your point earlier, had to kind of like map your data set to. Or could you maybe clarify a bit more how exactly this was done?
Yeah, so it was mostly done through R, which was very useful, but I had to download Free Surfer, which contained the distro atlas and the vertices that corresponded to each region. So I just kind of inputted them into R and then inputted the model, the linear discriminant analysis model. So it had the weights and then it had the names, but I had to clean the names because at first it was just printing white brains, like they didn't match the names properly. So I had to like run a lot of cleaning to correctly match the names, and once the names are matched, you can match the weights to the regions, and then you just ran some R code. It was some package, and it just produced the brains for you with the weights on them.
Makes sense. But in your data specifically,
Yeah.
You kind that you downloaded. You just have to make sure that those match. So how did you get the names?
Yeah, we used data from the Autism Center of Excellence, and that data set already had the volume names on it.
Understood.
Yes.
So those are consistent across the two data sets?
Yes, they were relatively consistent. They had maybe some extra like letters in the beginning that I just had to get rid of.
Super cool.
Thank you.
Any other questions?
Yes.
So the purpose of this really is to try to find a way to diagnose early without relying on behavioral differences to get around the bias that we're talking about?
Yes, that would be the end goal. Although I think in my head it would more support the behavioral based tests. I feel like there could be implications in just using this, so I feel like at least integrating both would be helpful. Thank you.
Awesome. Let's give thanks again.
Thank you for your time.